Welcome to this first tutorial for PECAn image analysis software. We're actually going to run our very first analysis using the PECAn Fiji plugin. So before we get started on the analysis, let's take a look at the example image we're going to be working with. And that can be downloaded from the GitHub repository. And once you've downloaded that from GitHub, go to the examples, click on sample images, and we're going to do example 1. So first things first, let's take this little image and open it in Fiji. And as you can see, this is sort of a multi-channel, multi-Z-plane image, so it's pretty complicated. For this example, this is sort of the standard experiment that we would do in terms of assessing cell competition in RPS3. So the green cells here are RPS3 loser cells. Blue just marks is DAPT marking the nuclei of all cells, and red is cleaved DCP1 marking dying cells. And so importantly, in terms of going into Fiji, is the order of channels. So if we look here, we've got channel 1, channel 2, and channel 3. Channel 1 is our DAPI signal. Channel 2 is our green signal that marks the patches of RPS3 cells. And channel 3 is our cell death marker. So we're going to keep all that in mind as we open up the analysis. So let's close all of that. Now let's launch PECAn. Go to the Plugins tab, scroll all the way to the bottom, launch PECAn. And so here we have sort of our options of how to analyze our images. And so this gives you all the basic sort of processes that you can do with the software. First thing is, do you want to use scaled units? So this is an important one in terms of your measurements. So if you launch, if you have an image that was generated on a microscope that automatically scales the units from pixels to microns or something, if you use this option, everything that you do in the in PECAn is going to be scaled according to whatever units your image is scaled to. So I keep that ticked by default. This next one is if you have like a stack, a 3D stack like the image that we just pulled out, but you want to project it down to a two-dimensional image, you don't want to deal with the three-dimensionality, you can this just gives you the option of doing a simple Z projection at the start of your analysis. The next thing sort of break down what kind of information you want to get out of your images. So here where it says, do you want to analyze the clones or genotypes within this image? This refers to a marker of the genotype of your images. In our case, that would be the green channel versus the unlabeled cells. So we do want to analyze that. This next one gives us the option to analyze individual cells within the image. Say if we have an option of looking at specific cells, we're going to do sort of more involved detailed analysis. This next one is, do we want to analyze discrete foci within the image? So this is any sort of discrete kind of blob of signal, sort of like the cell death marker that we just saw in the example image. Also, if we want to look at bulk fluorescence, we can tick this box here. If we want to do speckle analysis, we can tick this box here. And if our images are time-lapse or a video, we would tick this box here. So for the moment, we're just going to go with clones and genotypes and look at genotypes on their own. And press OK. Now this gives us sort of the detailed information of how we want to analyze our images. And if you want to get into the advanced parameters, you can click on these boxes here. We're going to ignore those for now. The first thing we're going to do is specify how we're going to choose our ROIs. By ROI, I mean the region of the image that PECAn is actually going to analyze and extract measurements from. So for this particular analysis, we're just going to do manual selections, which means that PECAn is going to open up the images for us, and we are going to go through and manually tell it where we want to look. And importantly, this option here which says specify ROI selection channel, this is sort of the channel that Fiji is going to use to select those ROIs. In this case, I'm going to be using the DAPI channel. And you see that it says we have a 1 here. That means the first channel, which is going to correspond to our DAPI channel. And you can just add an R or a B or any other sort of letter after that. That just specifies what channel, like what pseudo color we're going to do. So you can just leave it at 1 and it'll open up a default color. But I like it to be blue, so I'm going to press 1B. And this just gives us the option of how we're going to specify, how we're going to identify the specific genotypes within the image. And the default one, which can handle our example image pretty well, is this membrane tagged GFP option. This is this sort of step here gives PECAn a lot of its flexibility, and we're going to cover this in subsequent tutorials. But for now, just leave it at membrane tag GFP. And because we're looking at the second channel of the example image, just leave it at 2. And I want it to look green. It's not a particularly important step, but I'd like it to look green. Press G there. In this image, we only have one genotype that we associate with our clones that's green, so we're going to leave that as 1. And everything else can be left as is for the moment. If you tick this box here that says analyze each clone individually, we're just going to analyze each separate clone or each discrete clone as its own entity. So let's just press OK. And click on example 1. So PECAn will analyze any images that are in a given folder. Just put all the images of a given type with the same sort of setup of channels and stains and whatnot in a folder by itself with nothing else, and PECAn will by default do batch processing. So click example 1 and press select. Now the first thing it's going to do is open up this image for us. And this is our ROI selection step. So let's take a look at this image and think about where we actually want to do our measurements from. In the Padini lab, we like to sort of analyze this sort of flat central region here in the pouch. So I'm just going to draw it out. And then I can press finished, and that'll basically say that, tell the software that this region of interest is what we want to analyze. But we have the option of specifying different ROIs for different z-planes. So as you can kind of see, the shape of the pouch changes in 3D. So it works fine for z-planes 1, and it kind of starts to fall apart around 18 or 19. So let's say that this ROI is good for z-planes 1 to 18, and then we're going to add our ROI for another z-plane. And this gives us the option of adding a separate one. So it tells us at the top that we have z-planes assigned for z-planes 1 through 18. We just need to give it 1 for 19. And then we just tell it which z-planes to give it to, press finished, and we're done. Now PECAn is going to take care of the rest. And while we do that, let's take a look at what's happened in the example 1 folder. So you notice that we have our original example 1 folder. We also have at the same level a new folder that says example 1 process results. This is where all of our outputs are going to be put. And if we actually check the example 1 folder, we have some new entries in here. First off is a metadata file that just sort of tells you exactly what analysis you ran on these images in case you need to go back and check those things when you're writing up your method section. Also, this is a record of the selections that we gave to the to PECAn. So if we ever want to rerun this analysis on this image, we don't have to select our ROIs again. Those have been saved for us, and we can launch it again without having to worry too terribly much. And now as it's finished analyzing that image, it's going to put out all of its data files, save everything, and let us know that it's done. So first things first, whenever you're using PECAn, you want to check what the output image looks like. So to find that, just go to this process results folder, and you'll see the name of our image with a -- process stack. So we want to look at this to see exactly what Fiji did. So every measurement that the software does is going to be drawn out into the sort of panel that looks like this. And this panel is going to look different depending on the analysis that you run. So let's take a closer look. In this top left corner, we see our original image with the clone channel that we're analyzing, because that's the analysis that we did. And this white line here is the ROI that we drew. And you can kind of see, and it shifts at 19, the way we would expect. Over here is that same channel, except with a little outline telling us what has been marked as the clone regions. And you notice there's a central region here. In cell competition analysis, we really care about this, at least for RPS3 losers, we care about this concept of the border region. And so the border region here is the part that's between the two outlines. And the center region with the teal fill is letting us know what's the sort of center protected region in which competition isn't happening, or at least where cells are shielded from competition. And we can see this across Z-planes. And just up here on the top right, we just have a mask. So I'm pretty happy with that. I think it's doing a good job of identifying the RPS3 cells in the pouch region. Another thing is if you look down here, we tick that box that said individual clones analysis. So you'll see that these are distinct. Not all of these clones here overlap with each other. So here they're being treated as separate entities. And it tracks them in 3 dimensions, so if any of these regions sort of split or merge, you see that down here these guys aren't touching each other, but they touch each other later on. And now they're treated as one thing. So it accounts for mergings and splittings, and it allows you to look at each individual clonal region within your images. And again, here's the same thing, except it's an overlay. So you can see that's your clone 7, your clone 5, your clone 3. And all of this information has been spat out in terms of some CSV files. One, the clone tracking analysis gives us the individual analysis for each of these discrete regions, and the whole disk analysis is sort of the overall level of clone coverage. So we can open that and take a look at it and see that tells us the file name and the image name, and it gives us sort of the area of the ROI that's not the overall ROI area, the area of the ROI that's not clones, the clones area, the border area, and the center area. So that's a really basic analysis, just looking at sort of clone coverage. But we can go back to this image and do some fancier things with it. So if we launch PECAn back up, and let's say we want to analyze discrete foci. Let's add that option onto it. Press OK. And now you'll see that we have more information on this panel now. And all this information here is the same for our clone analysis. That's the same stuff we saw last time. But we want to tick this little box here. So we restore save ROI selection. So we don't have to do the manual selection again, provided that we're happy with the selections that we've done previously, which are now saved with your image. But let's analyze our images for discrete foci. And so we're just going to go with the default analysis. The default one works pretty well for things like cleaved caspase stainings, cleaved TCP1 stainings, or like a tunnel staining, just anything that's kind of a roughly cell-sized blob. This analysis works pretty well for. And then again, we just have to specify the channel. In this case, it's the 3rd channel. I want it to be to be red, so I put a little R next to it. You can totally ignore the R; it doesn't matter. But I just like the output colors to be a certain way. And what this is going to do is it's going to analyze the area of the image that's covered by this stain in the 3rd channel. If we want to count those as discrete entities, because that's sort of typically how we do it for cell death stainings, click this button and it will do an analysis of the individual foci and events. So then we just press OK. Tell it which folder our images are in. And it's going to launch right into it. You'll notice it just skipped the initial ROI selection step because we already told it which ROIs to use. And then we let it run for a short period of time, and it'll spit out its results for us. And how long this analysis takes is going to depend on a couple of things. It's going to depend how complicated your images are, how big your images are, how complicated the analysis that you're doing is. Generally speaking, bigger images and more complicated analyses take a longer time. And also it's going to depend on your computer. I'm just doing this on a fairly standard, sort of my just sort of default work computer. So if you have like a dedicated image analysis computer, this will zip by really quickly. If you do it on a laptop, it might take a little bit longer. In that case, what I would recommend doing is just setting up all of your images to analyze and letting it happen overnight. Because it usually takes, for one of these sort of standard analyses, about a couple minutes per image. Okay, now that it's finished our analysis, let's take a look at the output image again and see what it looks like. And you'll see that a new row has been added here at the bottom. So everything on these top two rows is exactly the same as it was last time. But you'll see we have on the left just the raw channel of our DCP1 stain, marking our apoptotic cells. But we got some other ones down here. So in the bottom right, this is just a mask showing where the cell death stain is. So red marks the foci staining that overlaps with the border region, and blue marks cell death that overlaps with the, or foci staining that overlaps with the central region. And the border region, we just do that based on a distance that we do like 8 microns. You can adjust that anytime that you want to, depending on the needs of your analysis. Or if it's not important for your analysis, you can just ignore it. All those it's not going to influence your measurements otherwise too terribly importantly. You will notice that up here in the single clones analysis, there's also a mask showing that we do measure sort of the foci overlap with these individual clone regions. But if we look down here, this is the result of our single foci tracking. So if we zoom in real nice and close, each of these has a little target on it with a number. And red badges mean that the foci for the most part is in the border region. A blue badge means it's for the most part in the center region, and it'll track it over time. So let's go and give a good example of that. Go apically. Let's look at number 7 up here. Oop, I guess we can't track that one. Where's a good one that we can track? Number 1, see, since it's roughly in the same position, we're counting it as one cell for that duration. And it'll track it in 3D to prevent double counting and things like that. Now, all of this information is going to be updated in our output CSV file. So the analysis, the hard work of quantifying these things and doing statistical tests will be handled by the image analysis app that we'll cover in a separate tutorial. Now we can do even more fancy kinds of analyses using PECAn. We can just launch it again, press OK. And another thing to note is if you spent the time messing with these variables and getting them exactly the way that you want them to be, which can take some time when you're getting started, to make sure you don't forget them, what you can do is you can come down here and save your current settings. And then press OK. Give a name for your new settings. So let's say demo settings, and press OK. Now it's going to relaunch this whole thing for you. Just press OK. And let's mess with something. Let's just switch to a bunch of random things we don't care about. So then we can just click this little thing that we just clicked before, and demo settings is there now. Click it, press OK. And now it's gone back to the settings that we had saved. So that's a nice feature just to make sure you don't lose your progress after you've been spending time messing with your settings. But we're going to add an extra step here. Let's go back to the start. And we're going to do our single cell analysis. Oops. So in this front panel where it says, do you want to count and analyze individual cells? Tick that box. And it's going to give us a new extra row, which is how would you like your single cells to be segmented in your single cell tracking settings? Let's turn that on to default, which is the one that's kind of optimized for this example image. And again, we need to tell it what channel we are using to segment. In this case, we're using the membrane stain, the green one. But also, we're going to cross-reference that stain against the nuclear stain, just to cross-reference to make sure that we're actually detecting something real and not something artifactual. So we want to specify which channel our nuclear stain is in. And you can disable that if you just want to do your segmentation based off the single channel, but for now we're just going to go with one because that's our DAPI channel. And we want to analyze them for spatial and fluorescence properties. If you disable this, you're just going to get a raw count of how many cells there are. If you do this one, it's going to measure different things about each individual cell. And if you come down here to the foci things, if we want to analyze the foci individually as well, you can analyze their spatial and fluorescence properties. So this is a pretty big analysis. It's going to take a while to run. So I press OK. Again, go back to example 1. And it's going to launch and run through and take care of all those things. So this one's probably going to take a decent amount of time to run. So I'm going to step away from my computer for a second and come back. Okay, as you can see, the analysis is now finished. Let's take a look and see what it did with the single cell tracking. And you'll notice this looks similar to last time, but a bit different. And the way you can tell it's different is if you zoom in on the top left panel, you'll see all these little badges. And each of these badges represents an individual cell being tracked in 3 dimensions. And you'll see this sort of yellow line. Not every single cell has a yellow line on every z-plane. Each cell ultimately does have one. That's just showing you the distance between that cell and the border between the loser cells and the winner cells, or the border between genotypes. And each cell is individually color-coded, and you can follow them over time. So let's see that yellow one going through a couple of z-planes. Also, we have the same thing down here for our foci. And you'll see each of those is labeled with the same yellow line measuring the distance to the border. The white line is distance between that cell and the center of the ROI. And you can see that for your individual cells up here. Now all of that information is going to be output in single cell tracking CSV. So now you have all of this information, all these parameters that you can look at. So that brings us to the end of our first tutorial in the usage of the PECAn image analysis software. In the next one, we are going to look at how to analyze the data generated by this software to actually do statistical tests and generate graphs.